Hello students, today we will be going to study some of the important topics related to cell biology and today I am going to tell you some important topics that could be asked in very short type of questions, short note and long type questions also. So if you are preparing for any type of competitive examinations or undergraduate exam, or you are preparing for any type of PG entrance examination where your syllabus is carrying cell biology. So these questions will be help you out in preparing your examination. So let's move with the important topics. This is for actually a part second from cell biology portion. Part वन जो मैंने है सेल बायोलॉजी के कुछ important topics थे वो already पिछले videos में मैंने आपको share किए थे. So let's start with the part second one. So question you can see, I have already discussed 34 questions about the cell biology, and the 35th one is what is glycocalyx. So this question was asked in 2016 in some of the UG examinations. So this could be help you out. आप ये question आप prepare कर सकते हैं. Then question number 36. In question number 36, please look what they are asking. Endoplasmic reticulum was discovered by. तो यहाँ पे आपको साइंटिस्ट का नाम देना है that who discovered endoplasmic reticulum. Similarly, who discovered mitochondria. Then 38th question is dash gave the term mitochondria. तो यहाँ पे किसने mitochondria term दी थी ये पूछा गया है. Then the term nucleolus was coined by. दिस इज़ द एग्जामिनेशन. सो बेसिकली आपको जो भी सेल ऑर्गेनेलीज हैं उनके साइंटिस्ट जिन्होंने इनको डिस्कवर किया है उनके नाम एक बार प्रिपेयर कर लेना है. देन सिनेप्सिस ऑफ क्रोमोसोम अकर्स इन विच फेज ऑफ सेल डिवीजन. दिस इज अगेन एन इम्पॉर्टेंट वन. ये सिनेप्सिस जो है क्रोमोसोम की वो सेल डिवीजन की किस फेज में देखने को मिलती है. सो दिस इज़ अ वेरी शॉर्ट टाइप ऑफ क्वेश्चन कुड बी आस्क्ड. देन क्रॉसिंग ओवर टेक्स प्लेस इन विच फेज ऑफ सेल. यहाँ पे आप ये नेक्स्ट क्वेश्चन ये प्रिपेयर कर सकते हैं. दिस इज अगेन एन इम्पॉर्टेंट वन. देन टेट्राड फॉर्मेशन अकर्स इन विच फेज ऑफ सेल डिवीजन. दिस इज ऑल्सो एन इम्पॉर्टेंट वन. व्हाट इज रिडक्शनल डिवीजन. न्यूक्लियर मेम्ब्रेन स्टार्ट्स डिसअपियरिंग इन विच फेज. इसमें आपसे पूछा गया है कि न्यूक्लियर मेम्ब्रेन कब डिसअपियर होना शुरू हो जाती है. इन विच फेज इट स्टार्ट्स डिसअपियरिंग. Then define monosomy and what is autopolyploidy. These were also asked in previous year question papers. What is aneuploidy? Define chiasmata. Okay, this is again an important one. Then again, what is backward stage? This is again an important question asked in previous year questions paper. And what is CDK? CDK is a cyclin dependent kinase. Okay. Define cyclin protein. What is quiescent phase? Already discussed in cell cycle in previous videos. Then define S phase, that is synthesis phase of cell cycle. Differentiate between or difference between prokaryotic and eukaryotic flagella. Peroxisome is discovered by, as I have already told you कि जितने भी सेल ऑर्गेनेलीज हैं उनको किसने डिस्कवर किया या नाम किसने कॉइन किया है टर्म किया है वो आप एक बार देख लेंगे. नेम वन सेल्फ रेप्लिकेटिंग सेल ऑर्गेनेली. दिस इज अगेन एन इम्पॉर्टेंट वन. नेम सिंगल मेम्ब्रेन सेल ऑर्गेनेलीज. जैसे आपको पता है कि सेल ऑर्गेनेलीज जो है डबल मेम्ब्रेन होते हैं जो यूकैरियोट्स में प्रेजेंट होते हैं. तो उसमें वो ये पूछ रहे हैं कि विच वन इज द सिंगल मेम्ब्रेन cell organelle. Then what is sedimentation coefficient? It was already asked in previous year question paper, so you have to consider this also. Then define plasmid. Who gave the cell theory? Then next one is what is integral protein. So these are the integral membrane protein which are present in between the plasma membrane. And yes, a very important one, fluid mosaic model was given by which scientist. So this is again an important question. Then unit membrane model is given by bilayer model is of plasma membrane is given by. These three models are there in which fluid mosaic model is the important one, but you have to prepare three of the all. And then the question is define plasmodesmata. What do you mean by active transport? And similarly, you can learn what do you mean by passive transport also. Then cell wall is made up of, so you have to learn their chemical composition of cell wall and chemical composition of plasma membrane as well as. What is cystine A? Define dictyosomes. Who coined the term ribosome? Ribosomes are discovered by which scientist? Define leucoplast. What is oxysomes? Define F0 F1 complex. Which organelle is known as powerhouse of the cell? You know that you have been learning since school times also. So this is the simplest one. And define blepharoplast. What is 9 plus arrangement? This is a type of flagellar arrangement in eukaryotic cells. Nucleolus is discovered by. Differentiate between primary and secondary cell wall. This is again an important one asked previously in some of the papers. Define tonoplast. What are microbodies? These were also these questions were asked in 2017. These all questions were asked in 2017 in UG examination. Then write on secondary constriction. G0 phase, that is also known as quiescent phase. Heterochromatin, write short note on heterochromatin. Define sex chromosome. These were asked in 2014 in UG examinations. Write note on lamp brush chromosome. This was asked in 2019. Write note on sex chromosome and sex determination in plants. Please do learn this also. This is an important one. Sex chromosome and sex determination in plants were asked in 2017 and also in previous year questions papers. So in this you have to look at the question sex determination in plants. Please do write about the sex determination in plants only. Do not write in human beings. Okay, so basically आप सेक्स डिटर्मिनेशन प्लांट्स में पढ़ के नहीं जाते हैं, आप एनिमल्स में और ह्यूमन बींग्स का आपको याद होता है. सो यू यूज टू राइट एक्स एंड वाई टाइप ऑफ क्रोमोसोम ओनली. So please do learn about the sex determination in plants also. Then what are sex chromosomes? Again asked in 2019. Discuss various types of sex chromosomes. So repeatedly it is has been asked, like in 2014 it has been asked like define sex chromosome. Then in 2017 it was asked like write note on sex chromosome and sex determination. Then again in 2019 it was asked that write what are sex chromosome and discuss various types of sex chromosome. So these are the important ones. Since 2014 to 2019 it was asked. And then describe the structure and function of cell wall. It was asked in 2014. Write note on function of cell wall. So you have to learn both of these things, like structure of cell wall and likewise the function also. So they can ask its structure also or its function also. So you have to prepare both of them. So then the question in 2017 was asked like write short note on active transport across plasma membrane. Okay, then in 2019 it was asked like differentiate between active and passive transport. So you have to prepare cell wall, its structure, its function. Similarly plasma membrane, its structure and function, and transport across plasma membrane, that is active and passive transport also. Then next question is fluidity of plasma membrane was asked in 2017. The note was asked, means the question was about 3 to 4 or 5 marks. Then the next question is different models to describe the structure of plasma membrane, specifically fluid mosaic model. In 2014 it was asked सो प्लाज्मा मेम्ब्रेन के जितने भी मॉडल है, स्ट्रक्चरल मॉडल्स जो भी दिए गए थे, जिसमें से बेस्ट स्टडीड मॉडल इज द फ्लूइड मोज़ेक मॉडल। तो आपसे स्पेसिफिकली या तो ये मेंशन करेंगे कि यू हैव टू राइट अबाउट द फ्लूइड मोज़ेक मॉडल, or they will ask about to describe the different models, so you have to describe all of the models those were given for plasma membrane structure. Then again in 2016 it was asked that discuss most accepted model of plasma membrane with label diagram and write the function of plasma membrane. So fluid mosaic model was the most accepted model, and so you have to write here about the fluid mosaic model. So in 2014 it was asked, then in 16 it was also asked, write note on fluid mosaic model of plasma membrane. Again in 2019 it was asked. So fluid mosaic model is the important one. You have to prepare it along with its diagram, its function, its structure and all. Then in question number 98, describe Golgi apparatus with diagram. It was asked in 2019, and its functions were asked in 2019. So you have to prepare its structure and its function also. Then similarly, short note on endoplasmic reticulum along with its diagram. It was asked in 2014, and in 2017 the structure and function, both of them were asked. Okay, so you have to prepare each and every cell organelles given in your syllabus, its structure and its function also, along with their diagrams, label diagram. Okay, then in 2019 differentiate between SER and RER, means smooth endoplasmic reticulum and rough endoplasmic reticulum was asked, so you have to prepare it. Then describe structure and function of nucleus in eukaryote. It was asked in 2017. And then in 2019 its structure and function was again asked. Then in 2014 ultrastructure of mitochondria was asked, and what was the features make it semi-autonomous, this question was asked. So you just not have to prepare the structure of mitochondria or just function of mitochondria. The questions in second part that was asked, that what features make it semi autonomous. सो अगर क्वेश्चन जो है वो आपका 4 मार्क्स में या 3 मार्क्स है और 5 मार्क्स में अगर ये डिवाइडेड है, तो हाफ मार्क्स जो है ये फर्स्ट पार्ट के होंगे एंड सेकेंड हाफ मार्क्स विल बी from the second part. So you just not have to go through with the ultra structure and function also, but you have to write about the features that make it semi autonomous also. क्योंकि अगर आप सिर्फ माइटोकॉन्ड्रिया के स्ट्रक्चर और डायग्राम पे ही स्ट्रिक्ट रहेंगे, इसके फंक्शन में ही स्ट्रिक्ट रहेंगे, तो पूरे मार्क्स आपको यहाँ पे नहीं मिलेंगे. और अगर ये 8 मार्क्स का अगर ये पूछा जाता है, सो यू विल be going to be deduct remaining 4 marks. So you have to prepare or you have to write each and every part of the question. Okay, then the next question is ultra structure of mitochondria and its function. It was again asked in 2016. Then in 2019 note on ultra structure of mitochondria was asked. So you have to prepare well mitochondria, its ultra structure, its function, and why it is called as a semi-autonomous, what feature makes it semi-autonomous. So these all points you have to prepare for mitochondria, and similarly for the different cell organelles also. So the next question is describe mitosis in plant cell with well label diagram. So you have to prepare the cell division along with label diagrams. It was asked in 2014. Then again in 2016 it was asked, mitosis was again asked. Then in 2016 short note on crossing over and its significance was asked. So you have to just not write about the crossing over, you have to write its significance also. So half of marks were given in both the part, so please do learn its significance also. Then write a note on synaptonymal complex was asked in 2019. Then write note on mitotic poison. I have made it bold. क्योंकि बेसिकली माइटोसिस एंड मियोसिस जो है वो आप प्रिपेयर करते हैं एग्जामिनेशन पर्पसेस के लिए, या जो उसके अंदर सिग्निफिकेंट जो प्रॉपर्टीज़ होती हैं या सिग्निफिकेंट जो भी उसमें एक्शंस परफॉर्म हो रहे होते हैं, हम उसी को बेसिकली कंसीडर करते हैं. बट some of you have not heard about the mitotic poison, so you must be get confused कि what is this mitotic poison. So let me clear it कि when any chemical substance which interfere, or we can say that which disturbs the mitotic cycle, or it inhibits the mitotic cycle by inhibiting its spindle fibers or formation of attachment of spindle fiber, so that chemical is behaved as a mitotic poison. So you have to write about the chemical that is used or that can inhibit the mitotic cycle. Okay, it was asked in 2019. Then next question is differentiate between mitosis and meiosis. So you have to prepare well about the cell cycle and mitosis, meiosis, its significance, and the important functions or important features that takes place in these cell division. Then again in 2019, difference between mitosis and meiosis was asked. Then write note on significance. In 2019 it was asked. Similarly, write note on significance on mitosis was also asked in 2019 in UG paper. Then what do you understand by cell division? Give a detailed account and add note on its significance. So this question is partitioned into three parts, in which in first part you have to give the description about the cell division. Then completely आपको cell division इसको समझाना पड़ेगा. Then उसके बाद में आपको इसके significance देने होंगे. So it was asked in 2017. Then the question was asked in 2014 to explain the karyotype and its significance. Then function of chromosomes was asked in 2014. What are chromosomes? Discuss the chemical composition and structure of chromosome with well-labeled diagram was asked in 2016. So you have to consider about on chromosome also, its structure, its function, and its chemical composition. Then again in 2019, chemical composition of chromosomes were asked. Then the next question, distinguish between chromosome and chromatid, it was asked in 2019. Difference between telomere and centromere was asked. Metacentric and acrocentric chromosome, haploploids and monoploids were asked in 2017. Then what are duplication? Explain with a suitable example, give its significance. Then question was asked in 2014, define polyploidy, describe its types and roles in crop improvement. It is also an important one. Polyploidy is important question that was asked in 2014. Again in 2019, polyploidy was asked, and give its role in plant breeding or crop improvement. So polyploidy ka question either it could be asked separately, that define polyploidy or what is polyploidy, but if it is asked in long answer type of questions for carrying 8 marks, so you have to write its significance and specifically in plant improvement and plant breeding. Then question number 129, what is chromosome aberration? This is again an important one. Discuss the structural aberration in chromosome. It was asked in 2016. In 2017 also it was asked about the structural aberration. Then 17 may variation in chromosome number. So you have to check out that what they are asking. Are they asking about the structural aberration, or they are asking about the variation in chromosome number? Then again in 2019, chromosomal aberration for numerical basis it was asked. Then the next question is describe the structure and function of chloroplast in detail. Then question was asked about the oxidative photophosphorylation, PS1 and PS2 of chloroplast in detail. So it is actually in some of the syllabus it is not clearly mentioned about the oxidative phosphorylation, but as it is associated with chloroplast, so it was asked in 2017. So you can go through it also. Then tablet the difference between SER and REL means smooth endoplasmic and rough endoplasmic reticulum was again uh, it was asked in 2016. Then write note on lysosome, write note on peroxisome, then describe the structure and functions of peroxisomes. It was asked in previous year question papers. So please do learn about the various cell organelles, their structure, their functions, significance, and their well labeled diagrams, and about chromosome, nucleus, and chromosomal abbreviations are again a important one. So these were some of the questions from previous year question papers, and do prepare these things, and best of luck for your examinations. Thank you so much.